Nucleic acids, just like your proteins, also display levels of hierarchy in terms of their structure. We have the primary, secondary, tertiary, and some references would also mention of a quaternary level of structure for nucleic acids. The primary structure of nucleic acids, just like the primary structure of your proteins, refer to the sequence of nucleotides in one strand of a nucleic acid. Remember, a strand of nucleic acid has directionality. You always start the sequence from the 5' end to the 3' end. The 5' end has a free phosphate group. This phosphate group is not attached to any nucleotide. Meanwhile, the 3' end has a sugar group whose 3' OH functional group is not attached to any phosphate group. The secondary structure is brought about by the hydrogen bonding of the nitrogenous bases in one or more strands of nucleic acid. We are familiar with the double helical structure of DNA, and this falls under the secondary structure, laid out on a two-dimensional plane. When we talk about RNA, it also displays what we call stem loops. Stem loops happen in one strand of RNA wherein a sequence of nucleotides in one region display complementarity with a sequence of nucleotides in another region of your RNA strand. So the RNA strand folds on itself and creates this stem and loop. Other references would refer to this as the hairpin loop. Other RNA strands can also display what is called a pseudoknot. A pseudoknot consists of two hairpins, but one of it is within the other. So if we follow this strand of RNA, starting from this end, we see that we have half of the stem, and then we see the loop. After this first loop, we see another half of the second stem, followed by the second half of the first stem, and then finally the second half of the second stem. So this happens in one RNA strand, and this is what we call the pseudoknot. Remember, the secondary structure is just a representation of the three-dimensional structure of your nucleic acid on a two-dimensional plane. So when we talk about the three-dimensional helical DNA, that is already the tertiary level. Now, a double helical DNA assumes more than one conformation. We have actually three types of double helical DNA. We have what we call the A form, the B form, and the Z form. All of these three-dimensional structures arise from the nitrogenous base pairing from two strands of deoxyribonucleic acid. We will learn more about this in a short while. So again, we also have the stem loop. if we are considering the three-dimensional stem loop or hairpin loop in your RNAs, then we are talking about the tertiary level of nucleic acid structure, and the same goes for the three-dimensional pseudo knot. In addition to this, there are some terms used to describe three-dimensional or secondary structure in RNA molecules. So you are now familiar with the stem loop or the hairpin loop and also the pseudo knot. We have what we call an internal loop. An internal loop is composed of nitrogenous bases that do not form hydrogen bonds with each other in a stem part. So if you notice here, we have a stem in this RNA, but we have the adenine bases coming from two different regions of the RNA molecule, not forming hydrogen bonds with each other, and therefore creating this what we call internal loop. We also have what we call the bulge loop, or simply bulge. The bulge is composed of one or more bases that do not form hydrogen bond with any other base from another region of your RNA strand, thereby creating this bulge on one half of your stem. And we have what we call the junction or multi-branch loop. So this is a region in your RNA molecule where you find several stems meeting. So it's like, from the name itself, a junction. Now, don't be confused if you hear the term stem loop. You might be confused. You might confuse it with the hairpin loop. Some references would refer to the hairpin loop as just the loop itself and the hairpin part as the helix. Some references would refer to hairpin loop as this whole secondary or tertiary structure. So I think it's more important if you know what these structures look like. So the hairpin loop or the stem loop is composed of a loop plus the stem part or the hairpin part, which is also sometimes referred to as helix, because these pairings cause the RNA molecule to form helical structures. Finally, we have the quaternary structure. The quaternary structure in the case of DNA would be the classical example would be your DNA bound to the histones or the histone proteins. So in this quaternary structure, there is another biomolecule involved. In the case of your RNA molecules, one RNA molecule can interact with another RNA molecule, and they can also create the structures that we have learned so far, such as the hairpin loop or the stem loop, pseudo knots, the bulges, the junction, and so on. So these are the levels of hierarchy in nucleic acid structure.